In this video, I'm going to talk about what is sometimes referred to as vitamin B8, and that is inositol, and in particular, this myo-inositol right here, which is why this paper has it boxed, and it says down here that it is the most common isomer found in food and animal tissues. So all these hydroxyl groups can have all these different isomers, and it would still be called inositol, but we're going to be focused here on myo-inositol. And so myo might tell you one thing, and that is that it is produced in the muscles. And so one of the reasons why why inositol and myo-inositol in particular is not often considered a vitamin B, even though it's sort of quote unquote vitamin B8, is because, well, because humans can produce it. This is produced in the human body, so we don't need to get it from our diet like we do with the other micronutrients, the other B vitamins in particular. But I thought I would go through this a little bit because we will run into inositol in later videos, especially when we get to things like cell signaling, because it is used in cell signaling. It's what's called a second messenger in cell signaling. But anyway, we'll get into the mechanism here. And so it starts from D-glucose and through this hexokinase, which is the first step in glycolysis. So hexokinase being the version of this enzyme used in muscles, where liver uses one called glucokinase. And when I get to the liver, I will be talking about the importance and the differences between those. But for our purposes, all we're doing is phosphorylating this hydroxyl group here, so we end up with this phosphate on it, and we end up with glucose 6-phosphate. And from there, we have this myo-inositol 1-phosphate synthase, or MIPS, or MIPS for short. And that is essentially just moving this phosphate from the 6th position onto the 1st position here. Well, that's one of the things that's happening. Probably the more important thing that's happening is this oxygen in the backbone is going away. So this hemiacetal group here is it's being gotten rid of, I guess you could say. And so the oxygen is taken out of the backbone. And then we have this inositol monophosphatase, which is just removing the phosphate, and we end up with our myo-inositol. And so these putting on and removing of phosphates, I'm not going to go through too much, but I do wanted to talk about this myo-inositol 1-phosphate synthase mechanism, because that one is a little bit more interesting, taking this oxygen out of the ring structure there. And so here on the left, I have this sort of overall mechanism. And so we have here the hexokinase step, and we have here that monophosphatase step. And so everything else, is oxidation and reduction and everything, is part of that MIPS, that MIPS step up here. And so we see that we have this sort of oxidation, which does this ring opening, and then we have an enolization, which is sort of moving the double bond from this oxygen into the carbons there. Then we have an aldol cyclization, which is essentially, we're taking the double bond from here and having that attack that carbonyl carbon there, that aldehyde carbon there, so that we close the ring, and then we have another reduction, which then turns this ketone back into a hydroxyl group here. And so I have this more in-depth mechanism, and this comes from the same paper where they got the structure that I have right here. And so we can see in gray right here, this is the reactant here. This is that glucose 6 phosphate, or at least it's sort of a it's sort of something that's supposed to simulate the glucose 6 phosphate, so it fits into the active site in the same way. And then we have over here in yellow that is the NADH, and so we see the nicotinamide moiety right here, which is the business end of it, brought right in close to our reactant here. Then the magenta, in magenta here, are the active site residues that are sort of holding the reactant in here and performing the catalysis mechanism. And so those are the same residues that are numbered in this right here, in this mechanism right here. So this ring right here, that is the nicotinamide ring, which is in our structure here. That's this yellow ring right here. And so that is the business end of the NADH. And so this structure right here is our glucose 6 phosphate. So what you see is we have a oxidation going on here where this hydride is being extracted. So it's not a simple proton extraction like you would have in acids and bases. This is a hydride extraction. So the hydrogen is going along with two electrons, and so that binds onto this, producing that oxidation reaction. It's oxidizing this hydroxyl group right here, and so that is turning that into a carbonyl group, which is what we have right here. And then we have that enolization reaction where this shows one of the oxygens from the phosphate actually deprotonating this right here, and so the electrons for that are moving down to generate that double bond right there, where the electrons in the double bond for this carbonyl is deprotonating from one of these lysine residues here. And then so from there, what we have is so what we have from there is this lysine is then reprotonating this. So this oxygen is now protonated, and so now the lysine is extracting the hydrogen off of there. So the electrons from this oxygen are collapsing down, then the electrons from this double bond are going over and grabbing onto the carbon right here on the double bond to that oxygen, so that aldehyde carbon right there, which was produced up here. Well, it's already on there from up here, and so this is that carbonyl, or that aldehyde right there, and so it's binding onto this aldehyde carbon, and so we see this carbon here ends up bound to that carbon there, so we have a ring closure. But now we have this carbonyl right here, and so now we have the reduction step. So the hydrogen, the hydride that was pulled off in this first step right here, is now going and attacking the carbonyl carbon on here, and then so the electrons come up and extract a proton from this lysine here, and so we end up with our product right here, which is the myo-inositol. And so that is the mechanism for this this step up here, which is this myo-inositol 1-phosphate synthase. And so what we see going on here, once again, this first step, which is the oxidation, it's this oxidation right here, is this hydride being pulled off from our reactant on and put onto the NAD right here. So on the nicotinamide moiety right here. Then we have this enolization, which is generating this double bond between carbons and getting rid of the double bond on this oxygen here. But then the electrons from the oxygen collapse back down so that the electrons in this double bond can then go and attack the aldehyde carbon right over here. And so then the electrons from that will then extract a hydrogen off of this lysine. And then we have a reduction step right here. So with the same hydride on our NADH is attacking the carbonyl carbon here, and then reducing that carbon there so that we end up with our myo-inositol. And so from there, the inositol, when it's incorporated into sort of as a second messenger, will have all kinds of different phosphorylations and things like that, and be attached to other molecules, in particular onto different types of diglycerols. And so then it's incorporated into cell membranes, and then it can be used as a second messenger. But we will encounter that in later videos when I start going over more of the metabolism and biochemistry, and we get into things like cell signaling. But anyway, the takeaway message, I guess, is that we start with the glucose, which we get from our diet. So that's just sugar. Then this first step, hexokinase right here, which is part of glycolysis. And then this glucose 6-phosphate can get shunted off by this myo-inositol 1-phosphate synthase. And myo telling us that this is happening in the muscle. So you might have heard of things like myostatin and stuff like that, which are pathways in the muscles. But anyway, let's not get ahead of ourselves. So we have this myo-inositol 1-phosphate synthase, which removes that oxygen in the ring structure there. Then we have this inositol monophosphatase 1, which then removes that phosphate off of this, and we end up with our myo-inositol. But anyway, I don't want to dwell on this too long, so I hope you found this video helpful, and I will see you in another video.